In our previous video, we talked about lipids and their classification. In this video, we'll discuss about lipid metabolism. Lipid metabolism involves the synthesis and degradation of lipids. These two processes of synthesis and degradation are in constant state of equilibrium, which means some lipids are synthesized and others oxidized to produce energy. Digestion is the first step in lipid metabolism. First step of digestion primarily occurs in the stomach by the help of lingual lipase and gastric lipase. Lingual lipase originates from the glands at the back of the tongue, while gastric lipase is secreted from the gastric mucosa. Lingual lipase, just like gastric lipase, is most active at slightly acidic pH. That's why it is more effective in the acidic environment of the stomach than in the neutral pH in the mouth. Triglycerides containing short or medium-chain fatty acids are hydrolyzed by these enzymes, releasing free fatty acids and glycerol molecule. As infants do not produce sufficient amount of pancreatic lipase, so these lipases also play a role in lipid digestion in infants. Similarly, these enzymes also help in degrading thiacylglycerol molecule in patients with cystic fibrosis, in whom mutation in cystic fibrosis transmembrane conductance regulator gene leads to thick, sticky mucus that blocks ducts in the pancreas, thereby affecting enzyme release, ultimately leading to pancreatic insufficiency. Next step is emulsification of lipids in the small intestine. Emulsification is the process by which fats are broken down into smaller droplets, which allows for more efficient digestion and absorption. The process begins when the chyme, a mixture of partially digested food and gastric juices, leaves the stomach and enters the duodenum. Emulsification occurs by two mechanisms. One is by bile salts and the other is by mechanical mixing due to peristalsis. The liver produces and secretes bile into the duodenum. Bile contains bile salts, which are amphipathic molecules that have both hydrophilic and hydrophobic region. The hydrophobic region interacts with the fat droplets in the chyme, while the hydrophilic region faces towards the water-based contents in the small intestine. Lipids tend to clump together in the presence of water. This surface tension of lipid droplets creates a barrier that prevents digestive enzymes from interacting with the lipid molecules inside the droplet. Bile salts help to reduce the surface tension of lipid droplets. Peristalsis also helps to mix lipids with digestive enzymes and bile salts, allowing for more efficient digestion and absorption. There are two peptide hormones that play an important role in digestion. These are cholecystokinin and secretin. Cholecystokinin is produced from the mucosa of lower duodenum and jejunum in response to the presence of food, especially lipids and proteins. Cholecystokinin signals the gallbladder to contract, which releases bile into the small intestine. It also acts on the exocrine cells of pancreas, causing the release of digestive enzymes. It also decreases gastric motility, which slows down the movement of food from stomach to small intestine, allowing more time for digestion and absorption. On the other hand, secretin is released in response to acidity of chyme. Secretin stimulates the pancreas to stimulate bicarbonate release, which neutralizes the acidic chyme and helps maintain the pH of small intestine. Secretin also slows down stomach emptying so that the small intestine is not overwhelmed with too much acidic chyme at once. The triacylglycerol, cholesterol esters, and phospholipids are enzymatically degraded in the small intestine by pancreatic enzymes. Triacylglycerol molecules are large enough to be taken up by the enterocytes of the small intestine. Therefore, they are hydrolyzed by pancreatic lipase, which preferentially hydrolyzes long-chain triacylglycerol in contrast to gastric lipase. The primary products of hydrolysis by pancreatic lipase include 2-monoacylglycerol and free fatty acid. A zymogen procholipase, which is secreted by the pancreas, is activated in the intestine by the enzyme trypsin to colipase. Colipase has mainly two functions. It activates the pancreatic lipase. It binds the lipase and anchors it to the surface of fat droplets in the small intestine. Secondly, it prevents pancreatic lipase from being inhibited by bile salts. On the other hand, Cholesterol esters are hydrolyzed by cholesterol esterase, which produces cholesterol plus free fatty acid. In contrast to pancreatic lipase, activity of this enzyme is increased in the presence of bile salts. Pancreas also secretes proenzyme of phospholipase A2 that is activated by trypsin and requires bile salts for optimum activity. Phospholipase A2 removes one fatty acid from carbon number 2 of phospholipid, leaving a lysophospholipid. For example, the predominant phospholipid of digestion, phosphatidylcholine, becomes lysophosphatidylcholine. The remaining fatty acid at carbon 1 is removed by lysophospholipase, leaving a glyceryl phosphoryl base that is either excreted in feces, degraded further, or absorbed. In the next part, we will discuss further about the lipid metabolism. Thanks for watching.